We've made it to the last video lecture for blood bank, and in this last lecture of Unit 5, we're going to be talking about transplantation, immunology, and molecular testing. So we talked a little bit before, or we've kind of touched on the HLA system, and again, HLA stands for human leukocyte antigen. So this is the strongest immunologic barrier to successful allogenic organ and stem cell transplantation. Transplantation of a solid organ or hematopoietic stem cells into an allogenic host is likely to result in graft rejection or graft versus host disease in the absence of immunosuppressive therapy. And this HLA system, this is part of what makes it so hard to find a suitable donor for organ transplantation and why it takes so long, because it's so hard to find a perfect match. So HLA proteins are encoded by a set of closely linked genes located on the short arm of chromosome 6 within the major histocompatibility complex, or MHC. And we just briefly touched on HLA and MHC in Unit 1, so now we're going into more detail. So they're inherited as haplotypes, and if you remember that from Unit 1, that means a group of closely linked alleles that are on a single chromosome. The classical or transplant antigens consist of class 1 and class 2 proteins. So class 1 proteins in that HLA system include HLA A, B, and C. And then the class 2 proteins include HLA-DR, HLA-DQ, and HLA-DP. The HLA system is the most polymorphic genetic system in humans, and we used that term in microsome. So polymorphic just means presence of two or more different things. So in this case, it's two or more different genetic compositions among individuals in a population. So many HLA genes exist in the population, and there's a ton of different combinations of these genes that are possible in individuals. Okay, ABO blood group antigens are also critical when we're talking about organ and tissue transplantation, because these antibodies can bind to the corresponding antigens that are expressed on the vascular endothelium. That in turn can activate the complement cascade, which can lead to a very rapid rejection of the transplanted organ, which is known as hyperacute graft rejection. So organ transplants must be ABO compatible, just like with blood transfusions. And transplantation using incompatible donors has been achieved using plasma exchange and IV immunoglobulin administration. So those treatments help to reduce the ABO antibody to levels that lower the risk of that hyperacute rejection. And that hyperacute rejection occurs within minutes to hours after the vascular supply to the transplanted organ is established. Acute rejection can occur days to months after the transplant. Chronic rejection results from a process of graft arteriosclerosis characterized by progressive fibrosis and scarring with narrowing of the vessel lumen caused by proliferation of smooth muscle cells. And that's what, anytime you see the term or hear the term sclerosis, that's essentially what it means, is that there's been scarring that has resulted. Graft versus host disease. So this is seen in HCT transplants, less commonly in lung and liver transplants. Lymphoid cells in the graft mount an immune response against the host's histocompatibility antigens. In mismatched allogenic stem cell transplantation, the targets of graft versus host disease are the mismatched HLA proteins, whereas in matched stem cell transplantation, minor histocompatibility antigens are the ones that are targeted, that are the cause of that rejection. So some testing, histocompatibility testing. So first there's HLA typing. So you have to identify the HLA antigens or the phenotype or genes in a transplant candidate. So that's the recipient or the donor, and that's used to find the most suitable donor. So recipient, to find that most suitable donor-recipient combination from an immunologic standpoint. So, I mean, you can think of it in the same way that we think about doing cross matches and finding compatible blood products for a patient. But this is, when we're talking about tissue and organ transplantation, it's much, much more involved. Phenotypes or genotypes of the classical transplant antigens or genes are determined, and you can see the those options again there. Phenotyping is determined by complement-dependent cytotoxicity test, or the CDC test. So you've got T and B lymphocytes that are used for HLA class 1 typing. B lymphs are used for HLA class 2 typing. Binding of the antibody occurs only if the lymphocytes express the HLA antigen targeted by the antisera. And then after incubation with the antisera, rabbit complement is added if the cells have the HLA antigen recognized by the antibody in that well. Complement is activated and the cells are killed. And then dye is added and used to determine live from dead cells. So dead cells are able to take up the dye. They appear colored. Live cells cannot take up the dye, so they're colorless. And that's because their membrane remains intact, whereas the membranes of the dead cells are going to break down. Okay, genotyping is determined by PCR testing. So you've got ABO compatibility and infectious disease status that are also important considerations in donor selection. And then HLA antibody screening, identification, and cross-matching. Again, we're using either CDC method or flow cytometry. So the testing that we're talking about is really kind of the same concept that we're talking about for determining a patient's ABO status, their phenotype, and then cross-matching with compatible units. But like I said, the testing for HLA is just way, way more involved. There's just more to it. Okay, now we're getting into serological and molecular detection of bacterial infections. So first, there are antigen detection assays that are available to detect a wide variety of bacteria, viruses, parasites, and fungi in different clinical samples. Testing methodologies include several of these that we've already talked about: latex agglutination, enzyme-linked immunosorbent assay, which is your ELISA, lateral flow, immunochromatographic assays that detect the presence of an analyte, such as a bacterial antigen that may be present in a sample. And then several of these types of tests are going to be CLIA waived, meaning that they would be like moderate to low complexity and could be performed by someone in a doctor's office, like a medical assistant or maybe even a phlebotomist. Bacteria and viruses for which antigen detection is widely used include Strep pyogenes, Legionella pneumophila, rotavirus, respiratory syncytial virus or RSV, and influenza A and B. The most widely known molecular technology is polymerase chain reaction, or PCR, which we've talked about quite a bit previously, where you've got specific genetic sequences that are amplified and detected. And depending on the test or the thing that you're looking for, the thing that the test is looking for, you can have results available in as little as 20 minutes to a few hours. But when we're talking about some of those bacterial or viral pathogens or parasitic, that 20 minutes to a few hours is much, much faster than trying to identify it in culture. For some infectious agents, such as Neisseria gonorrhea and Chlamydia trachomatis, nucleic acid-based assays are widely available. They're more sensitive than those traditional culture methods. And then just generally speaking, molecular methods are going to be more sensitive than other methods, but there are not a ton of FDA-approved assays on the market, and the cost can be prohibitive. I know most hospital labs now at least have, especially since COVID, have means of testing molecularly for COVID, RSV, maybe strep and flu. And then labs also have tests for other molecular tests for other infectious agents too. So a lot of them have those respiratory panels, stool pathogen panels, panels that we can run for positive blood cultures, chlamydia, gonorrhea, C. diff, MRSA. So there are some that are available out there. But like the slide said, they can be very, very expensive to perform. Okay, serological diagnosis. So this has historically been what we've used to detect or confirm infections from organisms that are difficult to grow or for which other lab methods of diagnosing the infection are not available. So some of those organisms that this might be particularly helpful for: Anaplasma, Ehrlichia, Chlamydophila pneumoniae, Chlamydophila psittaci, Coxiella, Leptospira, Rickettsia species, and Trypanosoma pallidum. And generally what these tests are looking for is detection of either IgM or IgG antibodies, which would indicate either a recent or a previous exposure to an organism. And the antibody titer may be used to assess reactivation or re-exposure to an infectious agent. So, and I'm sure you probably talked about this in other classes, but the titer tells us kind of how much of it is present in the patient's blood. The primary disadvantage to this test method is that there's usually a delay between the start of the infection and the production of antibodies. Also, immunosuppressed patients may be unable to mount an antibody response. So even though they may be infected with a certain organism, if they're immunosuppressed, their body's not going to produce antibodies that we would then be, looking for, trying to detect in that test method. Okay, proteomics. So a proteome is the set of proteins expressed by an organism or a cell. Proteomics is the analysis and study of the proteins expressed by an organism's genome. Proteomics can be used to detect biomarkers, which are going to be, these are just biological molecules that are found in blood, other body fluids, or tissues that are signs of normal or abnormal processes or of a condition or disease. So this testing method is still pretty young, but best established clinical applications of proteomics so far are in the identification of markers for early diagnosis of cancers. And it also has the potential to be used to detect agents causing an infectious disease, monitor effectiveness of treatment, or identify changes in a disease caused by a microorganism or virus. Okay, so now we're going to talk about some of the testing methods that are available for different specific organisms. So with group A strep, culture is, of course, an option. It's just going to take a little bit longer. Detection of group A strep antigens, so this can be done with lateral flow immunochromatographic assays, which these are the type that you would expect to see in outpatient clinics, doctors' offices, urgent care facilities for rapid diagnosis of streptococcal pharyngitis. And this is the type that you guys performed in class. Although these assays have high sensitivities, cultures should be performed on negative tests. Those little cartridge lateral flow assay tests can take anywhere from 2 to 30 minutes, depending on the manufacturer of the test. But remember, we talked in micro about if that lateral flow assay, that rapid strep test is negative, that's when we would reflex to that culture. Okay, anti-streptolysin O or ASO testing, it is made to detect antibodies to the streptolysin O enzyme that is produced by group A strep. So if it's positive, this indicates a recent strep infection in patients suspected of having acute rheumatic fever or post-streptococcal glomerulonephritis after they've had that throat infection. ASO titers typically increase within 1 to 2 weeks after the group A strep infection, and they peak between 3 to 6 weeks after the patient initially presents with symptoms. So what we do is prepare dilutions of patient serum, add streptolysin O reagent, then we incubate, add reagent red cells as an indicator. So if enough antibodies are present, then the streptolysin O is neutralized and no hemolysis occurs. The titer is reported as the reciprocal of the highest dilution, demonstrating no hemolysis. The range of expected normal values depends on the patient's age, their geographic location, season of the year. The titer is reported in Todd units. A single ASO titer is considered to be moderately elevated if the titer is at least 240 Todd units in an adult and 320 Todd units in a child. And this is just an example, one of many different types of strep A tests that are available on the market, but it's generally what you're thinking about, where you've got that control line, that, the control line has to be present for the test to be valid, but then you've got the patient or the sample line there too that you're reading as positive or negative. Okay, H. pylori, we have ELISA and lateral flow assay methods available for antigen detection. Serological testing for IgG antibodies can be for ELISA, LA, LFA, and immunoblot available. So depending on the organism that you're trying to detect, a lot of manufacturers have both antigen and or antibody detection kits, but it kind of depends on what's more practical for the organism that you're looking for. So this is one that has antigen and antibody detection methods available. Mycoplasma pneumoniae, which is the leading cause of respiratory infections worldwide. So with antibody detection, IgM is the most useful diagnostic test because it likely indicates a recent infection. ELISAs are available for IgM or IgG. And the IgG could be positive if a patient has had a prior infection with the organism. Detection of cold agglutinins. So these are agglutinins capable of clumping red cells at 4 degrees Celsius, and that reaction is reversible when samples are warmed to 37 degrees Celsius. Cold agglutinins develop in about 50% of patients with Mycoplasma pneumoniae infections. This procedure, this methodology is not very sensitive or specific, so it's not really recommended as much anymore because it can also be seen in other conditions as well. And then we also now have molecular methods available. And the molecular methods I've seen have been where you're looking for Mycoplasma pneumoniae as part of a panel of pathogens. So this So, for example, the respiratory pathogen panel that's made for the BioFire Mycoplasma pneumoniae would just be one of maybe like 15 or 20 organisms that test is looking for. Okay, rickettsial infections. These organisms infect the endothelial cells, but do not circulate until the disease has severely progressed. There are several assays using real-time PCR that have been described in scientific literature, but there are currently no FDA-approved assays. So the serological gold standard method is IFA with Rickettsia rickettsii antigen performed on 2 paired serum samples to demonstrate a significant rise in antibody titers. And here, and really in a lot of testing, significant titer is considered a 4-fold increase in those antibody titers. And then the wild Felix, we talked about in micro, also is an older antibody agglutination test based on cross-reactivity of the patient's antibodies with antigens present on Proteus species, but this one again has low sensitivity and low specificity. Okay, spirochete diseases. So if you remember from micro, spirochetes are long, slender, coiled, gram-negative motile bacteria. Diseases caused by these organisms have a lot of similarities between the diseases. So that can include a localized skin infection that disseminates to different organs as the disease progresses. There's a latent stage and a cardiac and neurological involvement if the disease remains untreated. Treponema pallidum, if you remember, is the causative agent of syphilis. Typically spread through sexual transmission, but there are congenital infections that could be possible also. So the 4 stages of syphilis, you've got the primary stage where the initial lesion, which is called a canker, develops anywhere from 10 to 90 days after the infection, although the average is 21 days. That primary stage lasts 1 to 6 weeks, and the lesion heals spontaneously. Then the secondary stage is where you've got systemic dissemination of the organism about 1 to 2 months after the primary canker disappears. Symptoms of the secondary stage include generalized lymphadenopathy, malaise, fever, pharyngitis, a rash on the skin and mucous membranes. There could be neurological signs such as visual disturbances, hearing loss, tinnitus, and facial weakness appear in about half of patients with secondary syphilis, which indicates involvement with the central nervous system. These lesions persist from a few days up to 8 weeks. Spontaneous healing occurs here also, just like in the primary stage. Then the latent stage is characterized by a lack of clinical symptoms, and it gets divided into early late, which is less than 1 year, and late latent, in which the primary infection has occurred over a year previously. Patients are not infectious at this time except pregnant women who can pass the disease on to the fetus even if they are asymptomatic. And then the tertiary stage occurs most often between 10 to 30 years following the secondary stage. Tertiary syphilis has 3 major manifestations: systemic syphilis, cardiovascular disease, and neurosyphilis. The specimen of choice to diagnose this tertiary stage is spinal fluid. Okay, and then some of the direct detection methods. Dark field microscopy can be used to identify the presence of the organism Treponema pallidum in exudates from skin lesions. There are fluorescent antibody tests available. serological testing for the antibodies, and these are the ones that we talked about previously, that get divided into treponemal and non-treponemal testing. So the non-treponemal tests are the ones that are used as a screening tool, and then the treponemal would be the ones that are used for confirmation if you have a positive non-treponemal test. So the non-treponemal tests determine the presence of an antibody that forms against cardiolipin, which is a lipid material released from damaged host cells. So it's sometimes referred to as reagin. It's found in the serum of patients with syphilis, but also several other diseases. These tests are based on flocculation or precipitation reactions in which the patient antibody complexes with the cardiolipin antigen can be performed on serum, which is how most of the tests are performed, or CSF. If you're doing this specific test on CSF, you would have to modify the test a little bit for CSF. So essentially what the non-treponemal test is detecting is something secondary to the organism. It's not detecting the organism itself. So those non-treponemal tests include the VDRL, which stands for Venereal Disease Research Laboratory. The antigen for the test has to be prepared daily, and in that test mixture it uses 0.03% cardiolipin, 0.9% cholesterol, 0.21% lecithin. The other screening test that we talked about is the RPR, and you guys actually should have performed one of these in micro, I think. Of course, maybe you didn't. We might have done that in urines, but it looks like these little cards where you're looking for the tail. So the RPR results are reported as non-reactive, weakly reactive, or reactive. Any that are reactive or weakly reactive have to be titered. False positive results in RPR testing could be due to hepatitis infections, infectious mono, varicella, herpes, measles, malaria, tuberculosis, pregnancy, lupus, leprosy, IV drug use, autoimmune arthritis, advanced age, and advanced malignancy. So there are a lot of different things that can cause false positive RPR tests, which is why it's important then to get that confirmatory testing done. So the actual treponemal tests detect the antibody directed against the Treponema pallidum organism or against a specific treponemal antigen. So this one is testing for something that is actually based on the organism itself, not something secondary to it. And then the two that we talked about, two confirmatory tests that we talked about, were the FTA-ABS and the TPPA. FTA-ABS is one of the earliest confirmatory tests developed. It uses the Nichols strain of treponeme to detect Treponema pallidum, and it uses the Ryder strain to react with and extract non-pathogenic treponeme strains to prevent cross reactions. The TPPA test is just know that it's the other type of confirmatory test available. And then there is also molecular testing, or PCR, available for that one as well. And again, this is what the RPR test card looks like. So you're mixing a drop of, and these are the, this is the QC. So you've got a reactive, a weak reactive, and a non-reactive QC sample there to compare your patient samples to. So, and this is, they've actually, this is an undiluted patient sample, and then here they're doing the titers. So they've got serial dilutions of 1 to 2, 1 to 4, 1 to 8, 1 to 16 to see how far out they're getting a positive reaction. But you're missing mixing patient sample with the charcoal mixture, and you're looking for, you can kind of see it right here, what they're calling the tail or the speckled pattern there. Okay, viral infections. So viruses are submicroscopic pathogens. Size of a virus is measured in nanometers, so they're tiny. We can't see them with a normal light microscope. Their basic structure consists of a core of DNA or RNA packaged into a protein coat or capsid. In some viruses, the capsid is surrounded by an outer envelope of glycolipids and proteins that comes from the host cell membrane. Viruses are obligate intracellular pathogens that rely on the host cell for their replication and survival. Okay, so getting into some specific viruses, hepatitis causes inflammation of the liver. It can be identified as either early or acute stage of hepatitis, which the early or acute stages are characterized by just general flu-like symptoms, mild to moderate pain in the upper right quadrant of the abdomen. Progression of the disease leads to liver enlargement, which is referred to as hepatomegaly, and tenderness, jaundice, dark urine, light feces. And then the primary hepatitis viruses include hepatitis A, which is transmitted by fecal oral route, close person-to-person contact. This one is an RNA virus. Symptoms are usually self-limiting and resolve within 2 months. The test for anti-HAV IgM antibodies, which are detectable at the onset of symptoms and decline within 6 months. So that the IgM, again, is going to look for a more acute infection, or they have tests for anti-HAV IgG antibodies, which persist for life. So that would tell you more about if the patient has had a previous infection. And they do a lot of times, like hepatitis B is the big one that they want you to get vaccinated for if you're going into healthcare, but this is another one that may be recommended for healthcare workers. The hepatitis E virus is transmitted by the fecal oral route. It's a single-stranded RNA virus. It's also a self-limiting illness with recovery in 4 to 6 weeks in most people. There's an IgM anti-HEV antibodies test, which again is going to indicate an acute infection. That IgM level can remain elevated for about 8 weeks. And then the IgG anti-HEV test, so the IgG antibodies start to develop at about 4 weeks, and they're going to persist for years. So positive IgG is going to indicate a later stage of infection or the previous exposure. Okay, hepatitis B is transmitted through blood and other body fluids. It's the major cause of morbidity and mortality throughout the world. Is not the major cause, it's a major cause. Though most infections recover on their own within 6 months, and patients develop immunity to the virus. A chronic infection, which is defined as over 6 months, occurs in the majority of infected infants, about 1/3 of young children, and 10% of infected adults. And it's more common in immunosuppressed individuals. This one is a DNA virus. Testing for this one, so you can do for the hepatitis B surface antigen. That's the first marker to appear anywhere from 2 to 10 weeks after exposure. Levels are going to peak during the acute phase and decline as patients build those antibodies and then start to recover. The HBe antigen, may be elevated during a chronic infection, active during periods of active replication of the virus, and that one indicates high infectivity. So if the patient is positive for the HBe antigen at that time, then the infection could be more easily spread to someone else. And then the anti-HBc, which is the core antigen, IgM, again indicates a current or recent acute infection. IgG, anti-HB core, it starts being produced before IgM disappears, and then that one's going to persist for life, can be used to indicate a past infection. And then the IgG HBs is an indicator of immunity. And here's a chart that shows the different hepatitis that we just got finished talking about, at what points those test results are going to peak, and at what point they're going to, IgG versus IgM, how they're going to persist. Okay, hepatitis D virus, which is also known as Delta hepatitis, passed through parenteral route, can occur only in the presence of hepatitis B. So hepatitis D needs hepatitis B for its replication and expression. So if the patient has hepatitis D, they are also going to have hepatitis B. So it can be contracted as a co-infection with hepatitis B or as a superinfection in people who are already chronic HBV carriers. This one is an RNA virus. Testing for hepatitis D should be performed on all patients who are positive for the hepatitis B surface antigen. Detection of anti-HDV antibodies and HDV RNA are available. Hepatitis C, which is the one that most people have probably heard of the most, can be passed through parenteral route, with IV drug use being the main source of infection. It's the most common bloodborne infection in the United States. This one is a single-stranded RNA virus with the ability to mutate rapidly and variability, which makes developing a vaccine very difficult. Most patients are asymptomatic, with chronic infections developing in about 70% of cases. And then we can screen for anti-HCV IgG, which is detectable 8 to 10 weeks after exposure. It can remain positive for a lifetime, so it could detect a current or previous exposure. The CDC recommends the use of nucleic acid testing to detect HCV RNA for confirmation, which can be qualitative or quantitative. If it's quantitative, you could, I guess if it's qualitative too, either way, you can use that testing to monitor a patient's viral load in response to therapy. About 10% of patients can initially test negative. HIV, or human immunodeficiency virus, is the causative agent of the acquired immunodeficiency syndrome, or AIDS. It's caused by HIV-1 and HIV-2 viruses. HIV-1 is responsible for the majority of cases. It's more infectious. The majority of HIV-2 infections have occurred in West Africa. It's found to be less pathogenic with a lower rate of transmission. Transmission occurs through intimate sexual contact, contact with blood or body fluids. Those body fluids include blood, semen, vaginal secretions, CSF, synovial fluid, pleural, peritoneal, pericardial, amniotic fluids, saliva, which can happen during dental procedures, or this can also happen, be transmitted perinatally. The genome includes 3 main structural genes. So we've got the GAG, which codes for the core protein of the virus, the ENV, which codes for CD4 on T cells, and POL, which codes for enzymes necessary for replication. So the initial replication can be detected in the lab by the presence of the P24 antigen and viral RNA. So our lab testing that would be performed, HIV antibody detection, HIV antigen detection, viral nucleic acid testing, and CD4 T cell enumeration. The Epstein-Barr virus causes a wide spectrum of diseases, including infectious mono, lymphoproliferative disorders, and several malignancies. It's most commonly transmitted through salivary secretions. By adulthood, almost 100% of individuals have been infected. We can test for IgM and IgG, IgM anti-VCA, which for, even though we're testing for EBV, the VCA stands for viral capsid antigen. This is the most useful marker for acute IgM. IgG is also seen at the onset, but this one's going to persist for life. IgM anti-EAD can also indicate acute primary infection. Anti-eBNA appears during convalescence. Okay, this last little bit of the Unit 5, the last Unit 5 lecture is just going to be on molecular diagnostic techniques. And we did go through some of this in, it was in Unit 2, so if you need to go back and look at some of that as well, please do. So nucleic acids contain genetic information that codes for a protein structure. Primary characteristics of nucleic acids, such as nucleic acid complementarity and melting temperature, those characteristics are what form the basis of specificity for almost all nucleic acid-based tests. So this type of testing is very important in infectious disease testing because nucleic acids can be detected earlier than antibodies during the course of an illness. The 2 main kinds of nucleic acids are DNA and RNA. So DNA carries the primary genetic information within the chromosomes found in each cell. With RNA, there are 3 different types. You've got messenger RNA, transfer RNA, and ribosomal RNA, and then you've got non-coding RNA. The functions of these are in translating the genetic code of DNA into proteins and then in regulating the expression of that genetic code. So going back over a little bit of molecular terminology, nucleotide is composed of a phosphorylated deoxyribose or ribose sugar and a nitrogen base. Those nitrogen bases include, and we've talked about these before, adenine, cytosine, guanine, thymine, which are all found in DNA, and then uracil, which is found only in RNA. So in DNA, your base pairs that match are going to be adenine to thymine, cytosine to guanine, and then in RNA, uracil takes the place of thymine. The Human Genome Project was a research project that started in 1990. It was completed in 2003, where the complete genetic blueprint for human beings was mapped and they found over 3 billion base pairs. The chromosome is found in plants and animals. DNA is separated into multiple chromosomes per cell. Each chromosome is a double helix of DNA. So in humans, there are 46 chromosomes contained in the cell nucleus, 2 copies of each of the 22 autosomes, which are your non-sex chromosomes, and then 2 copies of the X chromosomes in females, or 1 copy each of the X and Y chromosomes in male. And then a haploid is 1 set of chromosomes. Diploid is 2 sets. Chromatin is the nuclear DNA with structural proteins. The gene is the basic physical unit of heredity. So sequences of nucleotides in chromosomes that carry instructions for synthesis of RNA, which when it gets translated into protein, leads to expression of hereditary characteristics. Gene expression is the process of transforming the genetic blueprint into functional products. An allele is a copy of a gene. It can demonstrate variations that determine a variation in a functional characteristic. Genotype is the genetic makeup. Phenotype is the outward expression of genes. So it's how the genotype expresses outwardly. A haplotype is an association of alleles at multiple loci. Mutation or variant is a change in the nucleotide sequence in DNA. Plasmid is an extra piece of DNA independent of the chromosomes and capable of replication. It's seen in bacteria and yeast and can be used to transfer genetic material from one cell to another. Protein synthesis. So there's a couple stages here. Replication involves the reproduction of DNA content from parent to daughter cells during cell division. Transcription is the conversion of DNA to RNA. Translation is RNA base sequence into an amino acid sequence. Polymerase is any of several enzymes that catalyze the formation of a long-chain molecule by linking smaller molecular units, such as nucleotides with nucleic acids. Okay, polymerase, we just said enzymes that catalyze the formation of a long-chain nucleotide by linking smaller molecular units. Endonuclease is any of a group of enzymes that degrade DNA or RNA molecules by breaking linkages within the polynucleotide chains. Restriction enzymes are endonucleases that will separate nucleotide bonds in DNA. They recognize and bind to specific nucleotide sequences in the DNA so that they will only separate the DNA at those specific locations. Amplification is a technique used to amplify the amount of the target or signal or probe so that specific sequences can be observed. So after amplification occurs, the product can be detected by different methods. An amplicon is the product of amplification or of the PCR cycles. The probes are short nucleic acids that bind to complementary sequences for the purpose of identifying sequences in patient specimens. And then the thermal cycler is an instrument used to carry out the amplification program. Okay, I know that was a lot of terminology, so just kind of look back over that and familiarize yourself with some of those terms. Okay, nucleic acid testing was designed to detect changes in the DNA sequence or to measure differences in amounts of RNA synthesized. 4 main approaches to nucleic acid analysis exist. So we've got the strand cleavage methods, hybridization methods, amplification methods, and sequencing. With the strand cleavage method, it was one of the first methods for analysis of DNA, was restriction enzyme mapping, restriction enzymes recognize palindromic sites. So a palindrome is something that reads the same front to back. So usually when you hear the term palindrome, you're talking about a word that reads the same front to back, but palindromic sites in molecular testing is how those base pairs, the sequence that they form. So a palindromic site would be like CTTC, and that's a very simple one, but that's an example. So enzymes are isolated from bacteria, so this serves as a primitive immune system. It allows bacteria to recognize its own DNA and degrade any incoming foreign DNA. DNA is characterized based on the pattern of fragments produced after incubation with restriction enzymes and electrophoresis. So this method is useful for investigating small genomes, such as those found in microorganisms or plasmids. And CRISPR technology, it's actually been around for a little while now, but we've been hearing about it more in some recent years. It stands for clustered, regularly interspaced, short palindromic repeats. So this is another type of restriction system found in certain bacteria. You've got repeated DNA sequences in microbes that are interrupted by spacer sequences matching regions extracted from invading plasmids or viruses. These spacer sequences serve as adaptive immunity with memory of invading DNA. So CRISPR has been used for DNA analysis, gene therapy, genome editing, and clinical applications for this technology are still under development. So, and I think that's a little outdated right now because I think they have come a long way with CRISPR technology now. Hybridization methods involves the binding of two complementary strands of nucleic acids. So that includes the template and the probe. In southern blot, you've got DNA that's cleaved into smaller fragments with restriction enzymes, separating the fragments by gel electrophoresis and identifying the region of interest through hybridization with labeled probes. In northern blot, it's used to analyze RNA structure and expression. It's mostly used for research and not for diagnostic purposes. Western blot, you've got a patient serum, a cell lysate or extracted proteins that are separated by gel electrophoresis and blotted to a membrane. The probes used are poly or monoclonal antibodies specific to the protein of interest. Then detection is performed with secondary antibody enzyme conjugates and color or light producing substrates. Western blot used to be the method of choice for detection of HIV and Borrelia burgdorferi, but it's been replaced by less labor-intensive methods, but Western blot is still used in research labs. Array methods, or microarrays, use thousands of targets with probes bound to a very small area, such as a microscopic slide or beads. The test sample is labeled and hybridized to those immobilized probes. And then for analysis, sample labeling is fluorescent, which allows for detection of the test sample and reference sample, which results in increased or decreased amounts of test material relative to whatever the normal reference range is. In situ hybridization is the detection of targets in place as they appear in tissues, cells, and subcellular structures, with labeled targets used to bind or hybridize to the targets. This method is used frequently in pathology studies of tissues and cell suspensions. Immunohistochemistry is a type of in situ hybridization that uses labeled antibodies to detect the presence of clinically significant protein targets, such as tumor cells. Probes for these tests are monoclonal antibodies linked to enzymes that produce visible signals from chromogenic substrates. Fluorescent in situ hybridization uses fluorescently labeled probes attached to complementary base sequences of target DNA found in intact patient cells that are applied to a solid surface and require specialized microscopes equipped to detect the emitted fluorescent signals. This is commonly performed to detect chromosome abnormalities, such as microdeletions and gene amplifications. So some different amplification methods. These most frequently used methods in molecular diagnostics involve some aspect of amplification during the testing method. So polymerase chain reaction, or PCR, is an amplification method. Involves an in vitro DNA replication procedure that includes the sample containing the DNA template to be copied, primers to prime the synthesis of the copies, the 4 deoxyribonucleotides, DNA polymerase to join the dNTPs, and a buffer with an optimal pH for the polymerase activity. The PCR reaction mix is subjected to an amplification program consisting of a designated number of cycles. That standard 3-step cycle includes a denaturing step, in which the DNA breaks apart or denatures into single strands as it is heated. So it gets heated to between 94 and 96 degrees Celsius, and this is when DNA breaks apart. And then there's an annealing step that binds the primers and the targets. This happens at between 50 and 70 degrees Celsius. There's an extension step where the complementary nucleotides are added to the primers to complete DNA synthesis. This happens at between 68 and 72 degrees Celsius. And the products of the PCR reaction are visualized by gel or capillary gel electrophoresis as the last part of that PCR procedure. In real-time PCR, each cycle is detected and quantitated as it is completed. So there's a fluorescent dye on the probe that's added before amplification that allows some type of optical device to analyze the sample as the cycle progresses, or after the reaction is complete, which is referred to as end point measure. Real-time PCR is able to produce qualitative and quantitative amplification. Transcription-based amplification, this is an isothermal process, so it does not require repeated heating and cooling like in PCR. It targets RNA rather than DNA. It's more applicable to the detection of RNA viruses such as CMV, HCV, HIV. Then we've got probe amplification, where the number of target nucleic acid sequences in a sample is not changed, but rather the primers are extended or bound into many copies of detectable probes. And then signal amplification, where you've got large amounts of signal that are bound to the target sequences that are present in the sample. And then finally, DNA sequencing uses the idea of sequencing or detecting the nucleotide order of the nucleic acids. So the chain termination or Sanger method is a modification of the DNA replication process that uses modified nucleotide bases. The first human genome was sequenced using this technology. Okay, that is the end of the Unit 5 lecture, and I know a lot of this information in this last little bit covering the molecular testing techniques. I know that's a lot. My advice would be just to familiarize yourself with the terminology, and then just kind of very loosely see how that term, and familiarize yourself with the terminology, then go back and read about the different types of testing methods again, and I think they'll make more sense. But we've made it to the end of blood bank now.